did not do that, but that's de facto what he did. He said, trust me, trust the science. He's calling himself science. It's a subtle move. But he's done with intention. Whether he was cognizant of it or whether it was unconscious, he was still doing it nonetheless.
I am very excited about a book I've been writing and have actually submitted for printing, and I'm very excited for the team that's helped us put that together. The book's actually going to be titled Moving Beyond COVID-19 Lies and Restoring Hope and Health to Humanity. And in this book, I actually document, cite, and reference some of my major concerns about what is actually causing what we're seeing around the world being termed turbo cancers. Some of the ones that are the most problematic, we've never seen such aggressive brain tumors before. These are called glioblastomas. And we're seeing university students being diagnosed and within just a few short weeks dying from these glioblastomas that are growing so fast inside the skull and brainstem that these individuals are losing their lives. This is uncharacteristic of glioblastomas or any brain tumors in years prior to the COVID-19 vaccines coming out. So some of the things that we're going to be presenting in that book are what it is I've uncovered along the way in research that helps to define what is a most likely probable cause for initiating such rapid growth of cells in the brain and brain stems and nervous systems of the human body to create a mass of cells they're going to call tumors. They're so aggressively and so quickly developing that it has been termed turbo cancers, both by oncologists, by medical researchers. So you'll commonly see that term being used since the inception and introduction of the COVID-19 vaccines in December of 2020. In 1956, there are two scientists. Their names were Dr. Stanley Cohen and Rita. Dr. Stanley Cohen and Rita were actually asked to take chicken embryos and next to their nervous system, the brain and spinal cord cells, they were asked to put snake venom from a copperhead, from a rattlesnake, and from a moccasin. They were asked to put the venom and just put it next to the nervous system cells of the developing brain of a chicken. And within 72 hours, they saw massive growth and accelerated growth of these cells. They actually could create, within 72 hours, what they called a rapid growing glioblastoma within 72 hours, which is insane in my opinion. They termed this invisible factor, I'm quoting from the research study in 1956, they called this invisible side effect of snake venom on nerve cells, they called it nerve growth factor. Now, if any oncologist watches this documentary or this filming, you know that in your studies and in every single research study you've ever read or published related to neurological tumors or cancers, they use the term nerve growth factor. This came up in 1956 from these two scientists in 1956 studying the effects of snake venom on nerve tissue of chickens. So they're the ones that came up with coining this phrase, nerve growth factor, an invisible side effect of venom. What's even crazier is, in my opinion, is if you look at who funded that study, the American Cancer Society funded that study. Now, I'd like to know why would the American Cancer Society, whose mission statement claims they are going to be funding research to find cures for cancer, why are they funding studies in 1956 to see how fast they can create cancerous tumors in the brain of chickens, nerve cells, within a short period of time, which is a few days? I find that very disturbing. The actual venom component found in this study that they used and squirted out of a syringe next to the chicken nervous system tissue is actually called snake venom phosphodiesterase. All right, so snake venom phosphodiesterase, they discovered, has this invisible side effect to create accelerated nerve cell growth that could be defined as a future brain tumor or glioblastoma in a human. But this was in chickens. Well, these same two scientists, 5 years later in 1961, are given money again. Except this time they want to see what happens when the snake venom is actually put next to developing mammal brains, organs, skin of those animals. And they want to know, what does it do? The snake venom component that they use again 5 years later in 1961 accelerates tumor growth on the skin cells of the animals, in the organs of the animals. They could develop kidney tumors, spleen tumors, pancreas tumors, breast tumors, cervical tumors, ovarian tumors. They could create tumors in any part of the body of the mammal using snake venom. And they called this invisible side effect of venom, they called it epidermal growth factor. Now, every single oncologist, cancer doctor knows these two terms. They just may not have a clue they were first ever coined, termed, discovered until 1956 and 1961. All right, so nerve growth factor, epidermal growth factor. The disgusting part of both of these scenarios I'm summarizing for you and all people at home. Snake venom phosphodiesterase is the venom component used from these snake's venoms to create these rapidly accelerated tumors to grow in any tissue in the human body. Do you know what's crazy about that? We have heard since December of 2020 that there's these rapid forming tumors all over the body of human beings after getting the COVID-19 vaccines. Do you know that the two people that just created the COVID-19 mRNA vaccines, their names are Katalin Karikó and Drew Weissman, and they work at the University of Pennsylvania? Do you know what in every research paper since 2009 until the vaccines went out, do you know that every research study they used to create their, quote, mRNA gene therapy shots, do you know what they used in it that they claim they can use to cleave or cut your RNA to do gene editing? It's written twice in every paper. Snake venom phosphodiesterase. The same snake venom component that Dr. Stanley Cohen in 1956 and his lab buddy Rita were using in both of those studies, being paid by the American Cancer Society to see if snake venom will create brand new, accelerated, fast, rapid forming tumors. So yes, you can absolutely pinpoint that most likely what's in these shots are creating rapid forming cancers to form in any part of the body, because snake venom phosphodiesterase, since 1956, we know has an invisible side effect of nerve growth factor to cause nerve brain tumors, central nervous system, brain, spinal cord, nerves, and it also has what's called epidermal growth factor as a side effect that can create cancers in any part of the human body, because humans are mammals, just like the study in 1961. So you've got to understand the history of where these were discovered, then go look and see, is there any correlation to these shots? Yes, there is a massive one. There's actually two things that's in common with both of these sets of research scientists, both one male, one female. In 1986, Dr. Stanley Cohen and Rita both received the Nobel Prize in Medicine for their discoveries that snake venom, funded by the American Cancer Society, can create rapid forming turbo cancers. Well, what did Katalin Karikó and Drew Weissman at the University of Pennsylvania just receive in October of 2023? The Nobel Prize in Medicine for, once again, using snake venom phosphodiesterase as a way to damage the DNA of human beings and RNA, or manipulate RNA in their mRNA gene therapy shots, as quoted in each of their research studies since 2009. The American Cancer Society funded those research studies in 1956. What's really sad about these research studies, I find, is that the NIH funded all of the research for Katalin Karikó and Drew Weissman to do these shots starting in 2009. And that is listed as the funder for each of these mRNA gene therapy research studies to develop these mRNA vaccines for COVID. So yes, I'm very, very concerned that they are using in the shots venoms like snake venom phosphodiesterase that is quoted in the Nobel Prize winners for making the shots. It's in their research studies since 2009. Just go look at them. It's very, very easy to see. That's not the only thing, though, that I'm concerned about, creating turbo cancers inside these shots. Did you know that on the NIH's website right now, everybody calls these shots mRNA COVID-19 shots? There's no mRNA in the shots. The NIH on their website, they have a whole document right now how we manufactured the COVID-19 mRNA vaccines. And it says we didn't put any of the SARS-CoV-2 virus in it, the virus responsible for COVID-19 disease. They said we didn't put any of the virus in there. It's spelled out on their documents. There is no virus, and it says there's no mRNA. It says we put only in the mRNA COVID-19 shots something called DNA plasmids. Well, if you don't know what a DNA plasmid is, you might want to go look at their document. A DNA plasmid is a little bitty circular piece of DNA that's been genetically engineered by scientists. And inside this little piece of genetic material, this little hula hoop, they say in the document on the NIH's website, we embedded the instructions to tell the person who receives these shots or plasmids to tell their body to manufacture the spike proteins of COVID. So they said we embedded inside this little piece of DNA a little piece of the spike protein gene. They even draw it out on the diagram and they show it to you. This is where the spike protein gene is in the plasmid. Well, I have a big problem with that. What's the spike protein genes of COVID? What were they discovered to be in April of 2020, 4 months into the pandemic? Research scientists in France actually received samples of the spike proteins of the SARS-CoV-2 virus. Imagine this. Imagine requesting to test the spike proteins of SARS-CoV-2, a virus going around the world, spreading by coughing, and somehow labs in China and the United Kingdom are able to ship bottles of the spike proteins to the French researchers. Where'd they get them? So they say they received these from the UK and from China, and then they ran the DNA analysis of the spike proteins. What did they discover and publish? They said the spike protein genes of COVID, this virus called SARS-CoV-2, they said it was most identical to three things, the spike protein genes. Now remember, this is important if you want to understand the turbo cancers with the COVID-19 vaccines or post those vaccines. You have to know what they put in the shots. It's DNA plasmids. The NIH says they put the spike protein genes in there. Okay, great. Well, what was it discovered that the spike protein genes of the SARS-CoV-2 virus were? It was only discovered to be three things. The first thing they said it was identical to was king cobra venom. And this part of its venom called cobra toxin, which is a neurotoxic venom that paralyzes your prey from being able to breathe or run away. The second thing that it was found to be identical to, the spike protein genes of COVID, was bungarotoxin. And bungarotoxin is crate snake venom proteins, also a neurotoxin. So the spike protein genes of COVID were found to be identical to two snakes' venom neurotoxin proteins. Then they said there was small little pieces of the three different variants of the rabies virus that it also found was similar to. Now, before anybody runs off on the rabies thing, it's really not that significant because I have since discovered that the rabies virus, they say, it also has spike protein genes in it or on it, which I never knew. I never knew the rabies virus also had spike protein genes on it. I'm not sure if the audiences know this, but the rabies virus spike protein is called RGP, rabies glycoprotein. And guess what they say about the rabies glycoprotein, which is its spike protein gene? They say it is homologous, meaning almost identical to snake venom components. And they write that over and over and over in research. So now you've got spike protein genes that all look identical to either neurotoxic venoms from the king cobra or the crate snake called bungarotoxin, or you also have these three little segments that it's most identical to, like also on the rabies virus, but the rabies virus also has on the outside of it spike protein genes that are identical to snake venom. Okay, well, you just learned since 1956, they've known that snake venom, in the presence of any cells in a chicken or in a mammal will create rapid forming turbocancers. Well, if now we take this information about the spike protein genes and we put that back inside the DNA plasmid that the NIH says is the only thing they put in the mRNA shots, notice they didn't tell you mRNA plasmids, DNA plasmids. The spike protein genes were found to be either cobra toxin, bungaro toxin, or rabies virus, which is also homologous to snake venom protein. So no matter how you look at it, they just embedded genetic material that has the spike protein genes identical to snake venoms. But what's the purpose of the DNA plasmids, the NIH says on their documents? They state, we inject the vaccinated for COVID with these DNA plasmids that will instruct the patient's body to manufacture the spike protein genes. Well, if they're snake venoms and snake venom proteins, guess what they now are telling you the human body will start manufacturing from these DNA plasmids, which are genetic instructions to tell cells to make something they don't usually make. Well, what's the problem now? If you've got DNA plasmids being injected into everybody around the world, 5 billion people. Actually, 5 billion people did not receive the mRNA shots. But if you received a Pfizer and Moderna shot, these are the mRNA shots. They know and publish the mRNA shots for COVID contain DNA plasmids only. So these are genetic instructions to tell your body to do something. They're telling your body to make these venom spike proteins in your body, and the body doesn't stop. It just keeps making them and keeps making them and keeps making them. Well, the more little bits of venom protein being produced in your body, what do you think is going to happen eventually? What happens when you get venom in your body that your body can detect? If a scorpion stings you, if a snake bites you, if a stingray stings you, if you get any of those things in your body, you get venom inside your body, there are complications. Venom only has toxic effects in the human body, in anybody. There's only two main reasons for venom in any creature ever. It's either to kill the prey or neutralize the prey so it can't get away, or it's a defense mechanism to keep someone away. So in any case, your body is now a bioweapon manufacturing facility. What's the primary thing they're using to manufacture from the DNA plasmids? They're banking. When the governments around the world started injecting mRNA vaccines, they were banking on the majority of people having two things in their body, inside their bowels and inside their bloodstream, that would take the DNA plasmids inside of them and then start manufacturing the instructed spike protein gene. So if you got a shot that was just full of king cobra venom neurotoxin DNA plasmids, you will start developing respiratory failure, heart failure, arrhythmias, because these are neurological side effects. If you were injected with a DNA plasmid that had a separate venom protein on there that caused blood clotting, guess what's going to happen in your body? You're going to start making blood clotting. And we're seeing that all around the world. What happens if you get injected with what's called phospholipase A2, which is a snake venom component also? If that's embedded in the DNA plasmids, you inject that inside of COVID-19 vaccine victims, their body's going to start producing a venom protein that for the snake helps the body it injected it into dissolve and tear down all of its organs, leading to multiple organ failure, which will eventually lead to death. And before that, organ transplants. And what are you seeing all around the world? So this is how I explain to people how it is the COVID-19 vaccines are leading to what we call turbocancers formations around the world in any tissue in the human body. All right, so what are the two things that they're banking on your body has inside of most people getting vaccinated that's going to help them with their bioweapon engineering with these DNA plasmids? They're banking on two things, that most people all around the world have E. coli bacteria in their body and yeast or candida in their body. So E. coli bacteria is their favorite bacteria that is easily programmed and engineered whenever it's introduced to a DNA plasmid. The bacteria and yeast will draw in the DNA plasmid after it's been injected inside of a vaccinated. Anywhere in the body where E. coli is, it'll draw in the DNA plasmid and immediately start spitting out whatever it's instructed to do from the genetic instruction of the DNA plasmid. So if there was cobra toxin in this one specifically and it got inside an E. coli bacterium in your bowel, that E. coli bacteria is going to start making cobra toxin venom. 1 second after another. And then as we eat sugar and carbohydrates, we feed that one E. coli bacteria that's now programmed and genetically altered to now make venom. And if you don't think bacteria makes venom, what do you think Botox is? Botox is bacteria venom. And it is deadly toxic to humans. And they publish that all over in scientific papers. All right, so if you have E. coli in your body or yeast in your bowels that draws in the DNA plasmid, they will immediately start manufacturing whatever it's been instructed to. And this has been confirmed over and over and over by an incredible scientist in Italy named Carlo Brogna and his research team. They have taken long hauler COVID symptoms, or patients who have been diagnosed with long COVID, took their stool samples, ran their stool samples to see are there any venom proteins being manufactured in bacteria or yeast in their bowels. And 100% of them have replicating snake venoms, starfish venoms, and snail venoms from the ocean inside their bodies, and bacteria is manufacturing them. So Carlo Brogna has been helping medical doctors around the world, like Dr. Chetty in South Africa, figure out how to cure his long hauler COVID patients or long COVID patients. And once you identify that it's bacteria or yeast that have been engineered now to manufacture venoms, if you just wipe out those bacteria and get them out of the body, those individuals finally can recover.
Up next in this shocking